Hi, Bio 30. This is chapter 19.1, the Hardy-Weinberg Principle, looking at calculating how many organisms in a population are each of the three genotypes. So we know that there are typically these three genotypes in a regular dominant recessive situation, and we will look at calculating how many individuals are in each of these categories in a population. So have your calculators handy. Please watch till the end and you will be on your way to do this simple calculation. I would suggest that you could even pause this video and try some of these calculations as we go through. We start with this little case study of the sage grouse because they have noticed that there is not random mating. So there is actually certain features on the male that the females are attracted to, and this is likely the case in all of the other species. So there will be a certain basically sexual selection that happens, and in the case of the sage grouse, what we notice, you can watch a few of these videos, but the girls like the long tail feathers that are bright in color, as well as the large air sacs in the front that are bright yellow, and basically the grouse does a bit of a mating dance, puffing out his chest and showing off his feathers, and the girls really like those features. You can check that out on the videos. That concept will come up later as we look at some reasons for the allele frequencies changing over time. We know that there's many variations physically, behaviorally, even reproductively. There are many changes in individuals over generations. And we know that variations that exist in a species give it that species diversity, and diversity is a good thing, meaning the gene pool has more possible genes in it, and that's always a positive because if there was a change in the environment, having a variety of organisms with different features is more likely to have some of them survive. So what happens with Darwin and Wallace's idea here of natural selection is a reminder from Bio 20 is that the variations that help them survive their environment means that those organisms will survive and reproduce and pass those features on to their future generations. So the sources of variation, some of them could be just variations that were a new mix because of sexual reproduction. They could be because of mutations. They could be because of crossing over. And so diversity over time is very evident. So adaptations are inherited traits that are helpful for survival. And when we look at humans, because there's few offspring and generations are very long in time span, makes it difficult to study humans. But we also know that the environmental factor can change some physical features as well, like dyeing hair or other things like that are done that weren't there naturally at birth. So these definitions, a species are those individuals that can mate and make fertile offspring. They belong in one species, and a population is a whole group of that species in one place and one time. So you'll hear those words used. Population sampling is where we take a random sample, hopefully random enough to include all ethnic, geographical, social groups like surveys try to do. And then we use that data to say that this is what, Canadians think, and only a few have been randomly sampled, but that's how population sampling is done as well. The word gene pool, you should know this, is all of the alleles that occur in that population, including all the variations that could be passed along. So when we look at the three blood types, the three alleles in blood, there's A and B, and O. And so there's an idea of when you're looking at any particular gene, we can look at all the alleles for that gene. So for this, basically hair color, I guess, the B, there is the dominant allele and recessive allele. So those are the alleles for these, whatever, warthogs or whatever. And the majority of this chapter is going to be about calculating using the Hardy-Weinberg equation. Hardy-Weinberg was talking about how we realized that even though people thought that the certain alleles, recessive alleles typically, could be eliminated very easily from a population, Hardy-Weinberg said that does not necessarily happen because there should be a very similar composition of genes year after year, meaning that there's often a genetic equilibrium unless there is some upset to that population, in which case it'll start evolving. But because we have, for example, recessive alleles hidden in there in heterozygous individuals, there's still that chance that even if you don't see any recessive individuals, crossing a few heterozygous ones will have those recessive alleles pop back into the population. So Hardy-Weinberg analyzed genotypes in populations, and the word microevolution is these little changes that will happen in allele frequencies. So you might have 25% homozygous dominant, 50% heterozygous, and 20% homozygous recessive. And over time, these frequencies might change. That would be microevolution. And they like to study this to see if they can determine if there are big changes, what the reason for that was. For example, if they're monitoring bison in Elk Island National Park, they might want to pick some kind of a gene like shape of horn, long or curly, and take a look at how many of those individuals of each phenotype there are to just monitor the population. So this list here, the 5 conditions to maintain equilibrium, this will be a very, very important list of 5 things found in many questions in your future. So take note about these. They are basically saying for equilibrium to be occurring in exactly the same frequencies occurring year after year, all of these have to happen. It has to be a very large population with random mating, with no mutations, with no migration in or out, and no natural selection. So you guys may have realized through this list that these are not possible. It is not possible for all populations in any area to be large. We already know with the grouse, as well as other populations, that they do not randomly mate. There is sexual selection. We know that mutations are going to happen, and migrations will happen. They will move, and there will be natural selection because some will be better suited to the environment than others. So knowing that this list is almost impossible, you may be wondering why we bother even analyzing it. And the reason is because when they see allele frequencies change, they go back to this list and say which of these conditions are not being met and what's the cause of the change. And this is something that is analyzed if, for example, if we go back to the bison, if they see that we have less and less of a certain curly-horned bison, they may go and say what is going on and what are the conditions, the Hardy-Weinberg equilibrium conditions that are not being met, and maybe there's something they can like manage in terms of the reason, so they can basically make sure that diversity continues. So it says here, even though we can't meet these conditions, obviously in nature, we can predict why the changes or the evolution is happening. And in Hardy-Weinberg Principle, we're looking at one trait only. So like if we're looking at the horns, we just look at that one trait and check how the allele frequencies, exist maybe once every year. This will tell us whether that population is evolving. So as we maybe look at the difference between genotype and phenotypic frequency, remember genotype would be these three possible genotypes, and we can state them as a percentage. If they found out that 15% were homozygous recessive for these bears that were looking for curly hair was recessive, they found out 35% were homozygous dominant, and 50% then would be heterozygous, because they have to equal 100% being one of these three genotypes. Now, some of you may be sitting there going, yeah, you can definitely tell which are curly, but these, the ones that are straight-haired, you would never be able to tell which of these two genotypes you have. And that precisely is why we will use a calculation to figure that out and get a really good idea of what those two genotypic frequencies are. If we're just looking at phenotypes, that's pretty easy. We can say 85% are the straight-haired and 15% are the curly-haired. That's pretty easy to tell. But if we want genotypes, that's going to be something that's more likely the information we want if we're monitoring the population. We want to know those genotypes. So when we talk about alleles, remember that if I have 3, 6, 9, 12, 15 bears here, you realize these 15 bears each have two genes, so two alleles for that gene. So because they each have two alleles, you have to realize that 15 bears then would have 30 alleles. And so just keep that in mind when we're talking about allele frequency. We're actually counting up of this whole group what percentage are dominant alleles and what percentage are recessive alleles, remembering that each individual has two alleles. So if you had 100 bears, they'd have 200 alleles. And so you could look at the frequency of each genotype. So that would be how many two homozygous dominants we have. So that would be looking at how many of each genotype we have. That would be genotypic frequency. Or they may ask you about the allele frequency. So the two alleles we have here are dominant and recessive, H for hair type. And so if they looked at allele frequency, okay, if they were looking at allele frequency, they'd say how many big H's do we have percentage-wise and how many little h's do we have percentage-wise. And that would be an allele frequency. Whereas if you're looking at the three genotypes, that would be the genotypic frequency. So I just want you to realize that there will be different questions looking at either genotypic frequency or allele frequency. And you guys will see how to use the calculation for both of those. This little mouse example, before we even start to calculate, they're saying that we had, for homozygous dominant, 72 mice out of the 200. Heterozygous, we had 96 mice. And for homozygous recessive, we have 32 mice, but you realize the mice that are dominant, you cannot tell their genotype apart. If they knew exactly how many they had, though, magically, then you realize that that's 36%. If 72 out of 200 were this homozygous dominant, that would be 36% of the mice. 96 of them being heterozygous would be 48% of the mice, and homozygous recessive 16% of the mice. This is impossible to calculate unless you use the Hardy-Weinberg Principle, like I said, because these mice look exactly the same, which is why we use the calculation. But if we were looking at these numbers that we just calculated, those would be genotypic frequencies, how many of each genotype we have, whereas allele frequency would be where you find out how many big B's in the population, how many little b's in the population. And in this case, it says there's 0.6, 60% of the alleles are dominant ones, and 40% would be recessive ones, realizing that a 0.6 and 0.4 equals 1. So that's 100% of them are either big B or little b. And these 36, 48, and 16 also equal 1 or 100% because of the three allele genotypic frequencies, there are only those three genotypes. So they have to equal 1 if you're doing it in the numbers like 0.48, numbers that are less than 1, they will add up to 0.1. If you're using them as whole numbers, 36%, 48%, and 16%, they'll add up to 100%. So when we look at the equation, this is the equation that we're going to use for Hardy-Weinberg calculations. Do not be scared. All we're looking at here is how many homozygous recessives there are, how many heterozygous there are, and how many homozygous recessive there are. So how many homozygous dominant, if I said that right, heterozygous and homozygous recessive there are, because that will be 100% of the population. And we're using P's and Q's to signify the dominant and recessive allele. I don't know why they picked P's and Q's. There was an old saying, mind your P's and Q's. I don't know if that's why they use that, but that is what we're using, is that the dominant allele is signified by a P and the recessive as a Q. And so when we're looking at how many dominant and recessive alleles in the population, we're looking at how many P's and Q's. And when you add up, like we did before, if it was 0.60% were P's, then 40% would have to be Q's, because that has to equal to 1. All the alleles are just either dominant or recessive. For this one, for the Hardy-Weinberg calculation, where it says P squared plus 2PQ plus Q squared equals all of the individuals, I want to show you where they came up with that. Now, it's simpler than you think. All of the eggs that females have either have a dominant or recessive allele in them in that population. All the sperm of the males in the population either have a dominant or recessive allele. And so when you do your very simple old Punnett square, then you realize that there'll be some in the population that are homozygous dominant. If you look over here, some of them will be heterozygous and some of them will be homozygous recessive. Now, you notice that it's about a quarter in this Punnett square that's homozygous dominant, a quarter that is homozygous recessive, and about half of them that are heterozygous. In comparison, this is how they will be found. So there will be typically double the amount of heterozygous individuals in any population compared to the homozygous dominant or the homozygous recessive. So this simple Punnett square is exactly how they came up with the formula of P squared, which is the homozygous dominant, plus 2 heterozygouses, plus the homozygous recessive, and plus Q squared. And so this is all of the individuals. Is the homozygous dominant ones is P squared, big A times big A. There are 2 heterozygous ones, and there is the homozygous recessive. Now, when you try to determine exactly how many of individuals there are of each, you realize that these look the same. They look dominant. The ones that are homozygous dominant or heterozygous look the same. So typically, probably 99% of your questions are going to always say, this is how many in the population I see that are recessive. And if you know how many are recessive, I will show you how to use this calculation to figure out how many are the other 2 genotypic groups. So it's as simple as that. We can see that from the classic Punnett square, Hardy and Weinberg said this is the 3 different genotypes, and we can calculate exactly how many are the other two groups by looking at the recessive group. So let me show you how we do that. Okay, so in this question, these are ones that are in your notes so you can follow along, but my suggestion is that you always write this formula down and then write down what is being given to you in the question. So remember that this is who is homozygous dominant is p squared, who is heterozygous, you're going to need to do 2pq, and who is homozygous recessive is your q squared group. So this is how this works in this question. It's not really asking you to calculate anything right now. It's just saying put everything where it belongs. If 21% are homozygous dominant, we know that we're going to put the 21 here. And you notice I'm putting 0.21 because they all have to be as in a number between 0 and 1 because they're adding up to 1, not 100. So the 21% is stated as 0.21 is your homozygous dominant. Then it says 49% are heterozygous. So you put your 49% here. And then it says that 30% are homozygous recessive. And for this question, there's nothing to calculate. They just want you to see that adding up these three groups, in fact, is all of them 100%, or in this case, they add up to 1 because they're numbers that are between 0 and 1. And that's how we will be using our information. When it says 65%, you'll state it as 0.65 as we put it in the right place in our formula. Okay, so that's just where to put everything. The first calculation that you have is this one. And follow along, you can see it's not hard at all. We always are probably going to be given who is recessive. So you're probably always going to be given, almost always, the q squared. And so it says 16% are q squared, so you put that in there. And what you can do with that, this is the big moment of how we can determine the other two groups. What you've got to do is, if you know q squared is 0.16, you simply take a square root of q squared, because that, my friends, will give you q. Now, to find out the q, you put 0.16 in your calculator, push square root, and you find out that your q is 0.4. Now, don't forget, back in the beginning, it said p plus q is equal to 1, which means all the alleles are either p's or q's. So 40% of the alleles are q's, then guess what? 60% are p's because they equal to 1. So 1 minus 0.4 is 0.6. I always put a box around this. It's kind of like my legend of information because, my friends, this is going to give you the answer to the other two genotypic groups. How do you do that? Simply, if you know that now that q is 0.4 and p is 0.6, to get p squared, it's just 0.6 squared. So your answer is 36% of the population are homozygous dominant. How do we get the heterozygous group? It's 2 times p times q. So if we do that, 2 times p times q, we find out that 48% of the population is actually heterozygous, 2pq. Add these up, you can try it out. 36%, 48%, and 16% will equal 100. That's all of the individuals. There's one more part of the question here. It says, we know we have 500 in this population. How many are heterozygous? And so simply you say, well, I know 48% are heterozygous. So 48% of my 500 is how we answer that part of the question. And we know that that's now going to be an answer of 240 individuals that are actually heterozygous. And you could do the same thing for the others. If they said how many of the 500 are homozygous recessive, you could multiply 500 times 16%. Are homozygous dominant, you'd multiply the 500 times 36%. So that is how we use Hardy-Weinberg calculation to find the genotypic frequency of all of the groups. Remember, these are genotypic frequencies, p squared, 2pq, and q squared, whereas p and q, that is always considered the allele frequency. So if you ever are given numbers and it says the allele frequency is, they're telling you P or Q. If they're giving you the genotypic frequency, they're giving you one of these three groups. So be careful if they're saying genotypic frequency or if they're saying allele frequency. Okay, the word frequency does not tell you what they're giving or looking for. It's what specific thing that they're asking. Okay, let's try one more. 9% of the population is has a recessive genetic disorder. And so that would be over here, 9%. 9 out of 100 is 0.09, RQ squared. So you guys know what to do next. Your first thing is to put your information in your calculate your equation. Put your equation down. Put your 0.9 where it goes, 0.09. If you square root that, square root Q squared, you're going to get Q. And Q is 0.3. So 1 minus 0.3 is 0.7. So we know P is 0.7. Put a square around that. That's your legend of information that you can use to get your other two genotypic frequencies. For P squared, it would be 0.7 times 0.7. 7 times 7 is 49. So that's 49% of them are P squared. 2 times P times Q is going to be 2 times 0.7 times 0.3. So that works out to 42% of the population. And you are done your question. 49, 42, and 9 will equal 100% of them. And so you know that 49% are homozygous dominant, 42% are heterozygous creatures, and 9% were homozygous recessive, as stated in the question. Alrighty, this next question is talking about these tobacco plants, and I included this because it's a really good look at what 200 plants would look like and how the 200 plants each have 2 alleles, which means there's 400 alleles in this picture. And if you were asked about this, you could count how many of the 200 plants are homozygous dominant, how many are heterozygous, how many are homozygous recessive. So you could count that in this population, or you can do it with some Hardy-Weinberg calculations. If someone said, I have 280 dominant alleles and 120 recessive alleles, if you put that over the 400 alleles that there are, that's how it would calculate to 70% Ps and 30% Qs, 70% dominant alleles and 30% recessive alleles. So it's just showing you here that you can do that with a calculation of like 120 plants out of 400 that are recessive, meaning that if I take a square root of that Q squared, I'm going to get my recessive allele. You can calculate it that way by counting the recessive plants, or you can actually count them all up and see that there's 30% recessive little g's. You could do it that way. And when we write down the formula here, you know the Q squared would be all the albino plants that are homozygous recessive. And if we know that our allele frequency for Q is 0.3, I can say 0.3 times 0.3 is 0.09. That means 9% of my plants are homozygous recessive, but someone went and counted them, and indeed there are 18 out of the 200 that are albino homozygous recessive plants, little g, and that is 9%. So no matter if you count them or calculate it, you're getting the same thing. And then you can go on to use the 0.3 and 0.7 to find out the other allele frequencies as well, and they will match up to counting them. So I've got a few examples in this PowerPoint that are answer keys to what you're working on in the notes. So do try them before you look at these answer keys. What I encourage you to do is always write out the formula first when you do these questions. And when you write out the formula, that will help you to focus in on what, oh, this is a duck question. Okay, focus in on what is given and what is required in the question. And so for this one, if you look at this one, it's, let me see. It's saying that, am I on the right one? Yes. Okay. So it says that 36 of the 36 out of the 100, so it's 36%, are q squared. So I'm putting that in the right spot. If I square root that, I'm going to get my q, and my q is 0.6, meaning my p would have to be 0.4. Put a square around that legend and use that information to go back and do p squared, which is 0.4 times 0.4, 16%, and 2 times p times q to get the heterozygous, which is 48. So 16, 48, and 36 will be 100% of your squirrels. This one is the easy one because there were 100 squirrels. They are not always 100 in the population. So you have to be careful this time. There's 200 squirrels in question 4. And you just want to make sure that when you write your formula out, if there were 8 out of 200, if they give you a number that's the recessive, remember we're mostly always starting with recessive because you can actually see those in the population. And these two always look the same, so that's what we're calculating for. But if they tell you it's 8 out of 200, you may have to put that in your calculator first and find out that in your calculator 8 out of 200 is 0.04. Then you can square root that q squared, because that is q squared, the 8 out of 200. Then you square root that to get your q, which was going to be 0.2. So sometimes there is this extra step of dividing to find out what your q square is before you square it. And then you'll find out your q, and then of course your p would be 0.8 because they equal to 1. And once you find your p and q, you can go back and determine both other parts of your genotypic frequencies. Okay, so there is sometimes that extra step in there. Here's some definitions that you can review with. Remember that p is frequency of that gene or allele, and they may in the questions use either gene or allele frequency for p. Gene or allele frequency for the recessive is q, whereas if they're talking about p squared, it's the homozygous dominant. If they're talking about P2PQ, it's heterozygous genotype. Q squared, homozygous recessive genotype. So these are all genotypes, whereas P's and Q's are gene or allele frequencies. So if you ever see gene or allele frequency, they're asking you how many are dominant alleles, how many are recessive alleles in your population, also known as P or Q. So just remember gene or allele when that comes up, so you have the correct information for the formulas. Sometimes they give you one of these parts of the formula. Sometimes they give you the P or the Q, and you have to use those directly. You don't have to calculate them then. If they give you the gene or allele frequency, you can put them right into the question and find out your answer. Okay, the next 5 here, the cystic fibrosis question is kind of a human example. So the numbers you see are looking at things like these high numbers like of thousands of people. In this case, it's saying that there's 1 out of 2,000 that has it. So you're looking at a big number, but not very many. So when we put our formula in here, realize that 1 out of the 2,000 is what we have to put in our calculator to get your q squared. And when you put it in there, it's this really, really tiny number. and you still have to square root that q squared. So you have to put it in your calculator first, get your q squared, then you have to square root that q squared, and you're going to get a q of 0.02, which is p of 0.98, because it's in human conditions, it's often a very small number that have it. So that q recessive allele frequency is really, really tiny. Then you can go and find your 2pq, which they're asking you for, 2 times p times q, and get your final answer of 0.04. Alrighty, there's some answers to textbook questions if you wanted more, but there's tons in your notes. I'm going to just look at 2 more before you guys go on your way and try these for yourself. Remember to always write your formula down. That's an awesome starting point. This tells you that 32% are blonde people, so I put my 32% as the recessive q squared. When I square root my 32% or 0.32, I get a q of 0.57. 1 minus that is 0.43, so I know my p is 0.43. Now I can go back and answer 2pq. 2 times p times q will end up being 49%. And that was the one part of the question. And the other part of the question says how many blondes would be in 500. And so you go back and say, well, the blondes were 32%. In 500 people, 160 would be blonde. So you're just looking for, in this case, they didn't ask you for p squared. So you're just looking to answer just what is being asked in the question. Okay, the achondroplasia says that it's autosomal dominant, and it tells you the allele frequency. Now, guys, when it tells you allele frequency, realize that is, and it says it's dominant, that is p. So sometimes they do come and tell you the allele frequency. If p is 0.02, 1 minus that will give you the q of 0.98. Now there you have all the information you need to do the other 3, to find out all of the genotypic frequencies. You've got the allele frequencies, p and q, so simply q squared, 96% are q squared. P squared is going to be 0.02 times 0.02, so that's 0.04%. And then we've got 4% is our 2pq, and there you have it. Sometimes they start out with allele frequencies. For the Huntington's question, it says, again, allele frequency was 0.03 of this dominant disorder. So remember, that's a P because it's an allele frequency. Gene or allele frequency is P or Q. So if 0.03 is P, 0.97 is Q. And simple to do 2pq and get 0.06 is rounded to 0.06 times 100, 6% are heterozygous. And q squared would be 0.97 times 0.97, which is 0.94. In 800 people, you just multiply by 800 to see how many are actually not going to have this disease, because remember that it's a dominant, so everyone who is homozygous recessive doesn't have it. Okay, 2 more before I have you do your own practice. Albinism is, we read out the formula here. It tells you that 29 out of 725 have albinism. So remember, put that in your calculator. That's 0.04 before you can square root the q squared. So square root of your 0.04 tells you it's 0.02 is your q. Now, if 0.02 is q, that means 0.98 is p. But in this case, they're talking about just the q, so you're only having to use that q as 0.2 to what did I do wrong here? I think this was supposed to say 0.2, pardon me. And so it's 0.2 and 0.8. That's my P and Q. There we go. And so 0.2 being the Q, we can then say, if you're saying how many albinos are there, remember albinos are Q squared. So you have to go with the 0.04 when you multiply by 40. You have to use the amount that's the Q squared times 40, 1.6 people, and it's a statistic, so we can say 1.6 people will be albinos. Okie dokie. This last one is where we're comparing two different conditions, Tay-Sachs and sickle cell anemia, to see which are more prevalent in the population. And they come out and tell you that in 800 people, 32 had the Tay-Sachs. So we know that that's Q squared, 32 out of 800. So when we put that in the calculator and square root it, have to square root it, we get a 0.2 for Q for Tay-Sachs, and they already came out and told you that the frequency for the sickle cell is 0.13. So if we compare the allele frequencies of 0.2 for Tay-Sachs and 0.13 for sickle cell, we can see that this is like almost twice as many Tay-Sachs. So that said, who would get the money for research? If you had to pick one, probably would be the one that's double as prevalent in the population. So you're just finding the Q for both of those. And then it says how many would have it in a group of 1,000. You multiply, remember Q squared is how many have it in the population. So you've got to go Q squared for each one, multiplying the Q squared times 1,000, and we find out that Tay-Sachs is 40 individuals and sickle cell would be 17 individuals, if you found out that. Alrighty, so keep on trying your equations, guys. I will post some answers. I know they're in the PowerPoint, but I have them all even more clearly laid out. If you look at the answer keys for page 17 to 20 on my post, you can check your work there. Give these a try. They are not hard to do. Good